Alright, welcome back. We'll go over chapter 14, blood. Alright, the blood is a mixture of cells and cell fragments and dissolved chemicals and hormones and different kinds of waste like carbon dioxide and oxygen that go to and from cells throughout the body. Now, the volume of someone's blood will depend on someone's age and their gender and their overall size, but it's approximately about 8% of the person's body weight. There are two main parts of the blood. You have the liquid part, which is the plasma, and then you have the solid part, which is called the formed elements or the cells. So liquid, plasma, the cells are the formed elements. Now, the percentages of all these components are fairly consistent when someone is healthy. The plasma is about 55% of the blood. The white blood cells and platelets are approximately 1%. And the red blood cells are about 45%. Again, these are for when someone is in good health, a normal, healthy person. Now, the percentage of the red blood cells in a certain sample is called the hematocrit. So the hematocrit is actually a blood test. Here's a graphic explaining the information we just went over. A typical sample of blood here. The yellow is the plasma, about 55%. This small, thin blue band represents the white blood cells and platelets, a little bit less than 1%, and the other 45% are the red blood cells. Alright, all blood cells, no matter if they're red or white or platelets, are all formed in the red bone marrow through a process called hematopoiesis. Now, that initial stem cell will become more and more specialized and will form various types of blood cells. Now, before you panic, no, I'm not going to ask something like this on a test. This is more for informational purposes. All blood cells start off as this, the hemocystoblast. As they become more specialized, some will become white blood cells, which are all these here. We'll talk about all of these here in the next few minutes. And some will become lymphocytes. We'll talk about these in a few chapters when we talk about the lymphatic system and the immune system. But all blood cells start off as this one cell, the stem cell. Alright, we'll start first with the red blood cells. They're also called erythrocytes. The prefix ERYTH is Latin for red, so erythrocytes, red cells. Now, these cells are flat and biconcave, which means they cave in on both sides, and are very, very flexible, which allows them to go in and out of blood vessels pretty quickly. Now, this flat shape, it almost looks like a donut with a hole filled in. This broad surface allows them to carry, or allows them more surface area to carry more oxygen. And the oxygen is carried to the cells in a pigment called hemoglobin. So hemoglobin is a pigment that helps the red blood cells carry oxygen. Alright, here are two different views of a typical red blood cell. From the top view here, this is like a round, almost like a donut with the hole filled in here. And if you look at it from the side, see how it caves in here and it caves in here. It caves into two places, so it's biconcave. That's what's called a biconcave disc. And this broad area is where you'll find the hemoglobin carrying the oxygen. Alright, here's another example of, or another illustration of various red blood cells in a blood vessel, and you'll see the hemoglobin attached to the pigment within the molecule itself. This is how red blood cells look on a typical blood sample, which is what we'll see in lab. Most of the cells that you see when you look at blood under a microscope, almost all of them are going to be red blood cells, just like this. When red blood cells are immature, they're called reticulocytes. And when they're that young, they have a nucleus, but as they get older, they lose that nucleus. So reticulocytes have a nucleus, mature red blood cells do not. And the reason why they do that is, as they lose the nucleus, they have more room to carry more oxygen. Because other than carrying oxygen, red blood cells don't really do anything. They don't divide, they don't make proteins, they don't do much other than carry oxygen. That's why they typically only last about 120 days. Okay. The rate of how red blood cells are produced is controlled by a certain hormone that's made by the kidney called erythropoietin, or EPO. And just like we talked about with chapter 13, it's perfectly fine to call this erythropoietin or calling it EPO. Exactly the same thing. Okay. Like I mentioned on the last slide, red blood cells only last around 110, 120 days approximately. As they get older, they become more and more rigid, and they need to be broken down because they can't do their job anymore at that point. So as they get older and they're broken apart, the hemoglobin inside is taken up by special cells called macrophages, and they're broken down into various types of pigments. The first pigment is a greenish color, and it's called biliverdin. For those of you that speak Spanish or French, verd is the word for green, so biliverdin is a greenish pigment. And over time, this greenish pigment will turn into an orange pigment called bilirubin. So don't confuse these two, because these two will get asked on a test somehow. So the first pigment is green, biliverdin, then it becomes orange, bilirubin. Alright, that's all for the red blood cells. We'll move on to the white blood cells. These are also called leukocytes, and this can be spelled with a C or a K right here. It doesn't really matter. Leuko is a prefix for that means white, so leukocytes, white cells. And the primary function is to help fight infections. Now, there are 5 types of leukocytes that we'll cover, and they all fall into one of 2 major categories: granulocytes and agranulocytes. Now, these 5 types of white blood cells will vary in multiple ways, either by the type of cytoplasm, the appearance of the nucleus, the color that they stain when you're doing a blood sample, and you need to be able to recognize them for your lab practical. And there are some things that are very easy to spot, and some cells are easier to identify than others, and there are 2 that are very commonly confused with one another. And I'll go over some clues to help you distinguish between those 2. Alright, we'll start first with the granulocytes. And these 3 are called granulocytes because they look very grainy. Their cytoplasm looks like they have bits of sand in the cytoplasm, so it looks grainy. So that's why they're called granulocytes. And these will typically only last about 12 hours or so. And the granulocytes include these 3: the neutrophils, eosinophils, and basophils. And I'll talk about each one of these here one at a time. For neutrophils, the nucleus can have between 2 and 5 lobes, or 2 and 5 sections. These are the first white blood cells that will reach an infection site. And once they get there, they will engulf or phagocytize bacteria or fungus or the smaller viruses. And these are the majority of white blood cells in a normal, healthy person. They're about 55% of all white blood cells. And here's how one looks. Now, these cells here are all red blood cells. You can see how the cytoplasm looks kind of grainy, kind of gritty. So it's a granulocyte and a multi-lobe nucleus. That is a neutrophil. Next one, eosinophils. This is given away pretty easily by how it stains. It stains a very dark red color. No other blood cell stains this shade of color, very dark red. The nucleus will typically have 2 lobes. Sometimes it's hard to see because it's so grainy in the cytoplasm. And these are the cells that will help fight against parasitic infections. And these aren't terribly common. They're less than 3% of all white blood cells, between 2 to 3%. But the dead giveaway for eosinophils is the dark red color it stains. Again, here are all the red blood cells around it. The bilobe nucleus, or 2-lobed nucleus here. The gritty or grainy cytoplasm here. And it stains a dark red color. Nothing else looks like this at all. So when you see a red staining cell like this, it has to be an eosinophil. Next one, basophils. For those of you that have allergies, these are the ones that you'll want to listen to. These are the ones that will give you the most trouble. Their cytoplasm stains a very dark blue, almost a dark purple color. None of the other blood cells stain like this at all. So eosinophils always stain a dark red. Basophils always stain a dark blue or a purple. Now, basophils will migrate to tissues that are damaged and release heparin, which will inhibit blood clotting. So more blood can get there to help fight the infection. And also will release histamines. This will promote allergic reactions. So those of you who have allergies, that's why you take antihistamines to combat this from being released. So if you have allergies, these are the reasons why, these cells. And these are how they look. A very dark purple, and a little bit hard to tell in this image, but it is a grainy, kind of gritty cytoplasm. But nothing else looks like this. So when you see the dark red staining color as eosinophil, you see the dark purple like this, basophil. Alright, those last three we just did, those are the granulocytes. Now we'll talk about the last two, the agranulocytes. And these two are often confused with one another because they can look similar. But there's one key factor that makes one really stick out from the other one. For some generic information, the cytoplasm does not have a grainy appearance. They are agranulocytes. A means lacking or without, so without grains. So they don't have a gritty appearance to them. Each type, and there's two, has one large non-segmented nucleus. And the two types are monocytes and lymphocytes. And again, these two are the ones most commonly confused with one another. We'll start with the monocytes. These are the largest of all the blood cells. These are between 3 and 4 times the size of a typical red blood cell around it. So its size is what gives it away. When you see this on a tissue sample, which we'll go over here in a second, the really, really big size gives the monocyte away. Alright, these are the cells that will leave the blood after a few days and enter blood tissues, or enter the tissues, and that's when they're called macrophages. And that's where they will engulf and digest bacteria, cellular debris, dead cells, and so on. So macrophages are monocytes that have left the blood and entered the tissues. And these are about anywhere between 3 to 9% of all white blood cells. This is how they look. Now the typical red blood cell, but look how much bigger this is compared to this. So if you can't tell what a blood cell is, look at the cells around it. This is easily 3 or 4 times the size of these. And one large non-segmented nucleus. So that's a monocyte. Lymphocytes, for these, the nucleus will take up almost all the entire cell. What you usually see is one big nucleus and a small ring of cytoplasm around it. These will play a major role with the cells, or with the body's immunity system, producing both T and B cells. And we'll talk more about these in greater detail in a couple of chapters. The T cells will attack virus-infected cells like cancer cells or tumor cells. And the B cells are the cells that produce antibodies. And these will range anywhere from 25 to about 33% of all white blood cells. And these are a little bit bigger than the red blood cells around it, maybe twice as big or so, but one big nucleus and a smaller ring of cytoplasm around it. So yes, these are bigger than the red blood cells near it, but not as big as the monocytes. And here they are side by side. And this isn't a Photoshop. It actually kind of looks like a heart in this sample. But compare this, and this is slightly bigger, maybe about 1.5 the size. But compare this to these. Monocyte much larger compared to the red blood cells than the lymphocyte is. So these two are often confused. That's the dead giveaway for the monocyte, is the huge size difference between that and the cells around it. Here are all the 5 types that we just talked about, side by side. The top row, the granulocytes, the neutrophils, the grainy or gritty cytoplasm, multi-lobe nucleus. The eosinophils, same thing, gritty nucleus or gritty cytoplasm, dark red staining color, and the basophils, the dark purple or dark blue color. And the last two here, the agranulocytes, lymphocytes, and the monocytes. Okay. The last type of blood cell we'll talk about are the thrombocytes, or the platelets. These are not considered a real complete cell. They are basically a cell fragment. So they don't have a nucleus. That's why they're not considered to be a typical cell. If you were to take a typical plate and drop it on the floor and break it, you get fragments of that plate. It's the same analogy here. You get fragments of a cell. And of course, the main function for all thrombocytes or platelets is to assist the blood with clotting. And because of that, their lifespan is only about a week and a half. It's only about 10 days. And this arrow here is on one platelet here. But if you follow the cursor, that's a platelet. That's a platelet. these small fragments. All of these are platelets or thrombocytes, and a very unique small size gives it away also. So some of these blood cells are easy to pick out, some are a little bit more challenging, but platelets are pretty easy. Here's all 6 together on one image. See neutrophils here, the gritty cytoplasm, multi-lobe nucleus. Eosinophils, more of a reddish color. Basophils, more of a dark blue or purple. Lymphocytes here, slightly bigger than the red blood cells, but monocyte much bigger. So don't confuse these two here. And then lastly, the platelet right there. So that's all of them, all on one image. And again, you'll need to be able to identify all of these on a lab practical. Alright, we'll move on to the blood plasma. The liquid part of the blood, it's about 90% water. And here's where you find the platelets and all the cells that are suspended in this plasma. The proteins that are found here, the plasma proteins, which is why they're called plasma proteins, proteins that are found in plasma, have a very critical role with the body's immunity system, the movement of water in and out of the tissues, and also with the clotting of blood. There are 3 types of plasma proteins: the albumins here, globulins, and fibrinogen. We'll talk about these one at a time. The albumins, this is what will help determine osmotic pressure. Hopefully you remember what osmosis is, the moving of water from high concentration to an area of low concentration. So this is how movement of water is regulated from tissues and body compartments. These albumins will help to regulate how that water is moved in and out. The globulins, this has many functions, but the key ones are to transport lipids and vitamins, but also with the production of antibodies. The formal name for an antibody is a immunoglobulin. And again, we'll talk about that when we get to the lymphatic system. And the last one, fibrinogen, has a key role in the clotting of blood, which we'll talk about here next. Alright, the 3-step process that will stop bleeding is called hemostasis. And this is a very generic overview. There are a lot more detailed steps that are involved here, but I wanted to break it down into the big 3 because we really don't have the time to cover all the details that happens here. The first one is called the vascular spasm. If you think of it, when you touch something very hot, you jerk your hand back as a reflex. It's the same kind of process here. Whenever vessels get damaged, the smooth muscle part of that vessel will contract as a way to help lessen the amount of blood that is lost. So it's almost as a reflex. When a vessel gets damaged or tissue gets damaged, it will try to close itself off so it doesn't lose any more blood than it already has. That's called the vascular spasm. The second step is the platelet plug formation. Is where you have platelets that are traveling throughout the blood start to attach to one another and to the damaged ends of the blood vessels. As more and more platelets come, note that plug is larger and larger and larger. At that point, you have what's called coagulation. That's the forming of the blood clot itself. Fibrinogen is converted into a very strong, very sticky, thready compound called a fibrin. These threads stick together and form basically a very large spider web that will collect other cells as they pass over it, and that will help form and seal the blood clot. Alright, these next 2 terms are often confused with each other because they are related, but they're not the same. The first one, thrombus, is any blood clot that forms abnormally in any kind of vessel. So a thrombus is just another term for a blood clot. When this clot breaks off and starts to travel away from that original site and travel throughout the body, that's when it's called an embolism. And these can be very painful and very serious. This can be fatal. A pulmonary embolism is a clot that is broken off from somewhere else and traveled through the lungs, or traveling to the heart, or traveling to the brain. So embolisms can be potentially very dangerous. So don't confuse these two. A thrombus is another term for a clot, but when that breaks away and starts to travel, that's when it's called an embolism. Alright, here's those 3 steps put with the illustration. This is taken from the book. Step 1, the vascular spasm. This tissue that gets damaged will contract so it doesn't lose any more blood. You get the platelet plug start to form, where you have platelets attaching to the damaged ends. As more and more platelets come by, they stick together, form that plug. Then you have fibrinogen being converted into fibrin, that really sticky thread that will help form the actual clot itself. Again, this is how it actually looks. This isn't an illustration. This is a real image taken from a blood sample. Red blood cells here, and these things look like a big spider web are the fibrin threads. So they form a big lattice work, very large mesh, and cells as they travel by in the blood get caught up in here. And that's what helps form a clot on the damaged area. And this is a much more detailed, much more complex clot. All these individual things are fibrin threads. So again, that's a real image, that's not some illustration. Alright, we'll move on to the various blood types that people have. There are 4 blood types, type A, type B, type O, and type AB. The most common is type O. About 50% of people in the world are type O. And the least common is type AB. Now, what determines the blood type a person has is the presence or absence of a certain molecule on the surface of the red blood cell. Alright, we'll talk about the surface antigens of these red blood cells. This is how the body will recognize its own cells from something that is foreign. And every blood type has a very particular type of surface antigen. And it's fairly easy to keep straight. Type A blood has antigen A. Type B blood has antigen B. Type AB blood, antigens A and B. And then type O, no antigens. So you always think of type O as a 0 for no antigens. So these are very easy to keep straight. These are the antigens. That's often confused with this next phrase, antibodies. So don't confuse antigens with antibodies. Every blood type has specific antibodies to help protect itself from what's considered foreign blood. That's why when you get a blood transfusion, you can't just grab any random bag off the shelf. This is why blood types have to be matched correctly. Type A blood has anti-B antibodies. Type B blood has anti-A antibodies. Type AB has no antibodies at all. And type O blood has anti-A and anti-B antibodies. So don't confuse this antigens list with this antibodies list. Very often confused. Like I mentioned before, if you were to just grab some random bag of blood and get during a transfusion, the red blood cells will see it as foreign if it's not the right blood type, and it will attach or attack it, and those cells will clump together. And that process is called agglutination. And that's where you have the antigens and antibodies interlocking, and the red blood cells will be clumped together and basically torn apart. When this happens, the blood will become very thick, very viscous, and it's harder for the heart to pump the blood throughout the body. So this is very, very dangerous. That's why matching blood types has to be done correctly. The type O blood, or people who have type O, are called the universal donors because they can give blood to anybody because they have no surface antigens. So whoever they give their blood to isn't seen as foreign by that recipient. So type O's can donate blood to anyone, universal donors. So O and donors, type O. And people who have type AB blood are called universal recipients. They can receive blood from anybody because they don't see any type of blood as being foreign. So type O, universal donors. Type AB, universal recipients. This image is taken from a textbook. It summarizes what we just talked about with the surface antigens and the antibodies. If we're putting type A, type B, AB, and type O. Here's a graphic illustration of what agglutination looks like. Them clumping together, then they get torn apart. And this is how it actually looks. This whole image here should be red blood cells, the nice round oval shape, but instead they're all clumped together. What's left of them. So this can be very, very serious if not corrected quickly. Alright, next topic we'll talk about is the Rh blood group. It's called Rh because it's named after the rhesus monkey, where it was first studied. Now this is determined by a specific antigen on the surface of the red blood cells. So if someone has that antigen, that person is called Rh positive. So O positive, A positive, B positive, AB positive. That's what that positive or negative means. If you have it, you're positive. If you don't have it, you're negative. And a great majority of people have the Rh factor. So most people are going to be O positive, A positive, B positive, AB positive. It's much more common to be positive than negative. That's what that represents. Do you have that surface antigen? Yes or no? Now this Rh factor is an inherited trait and can cause problems during pregnancy. And here's the example. If a mother is Rh negative, but the baby is Rh positive, the woman's immune system will see the fetus as being something foreign, and it will start to attack it. And this usually isn't a problem during the first pregnancy, or during that current pregnancy. Usually isn't. But it can be very serious for any future pregnancies. So if that woman were to get pregnant again, there are already enough antibodies in her already to attack that next fetus. And this can lead to something called hemolytic disease of a newborn, which can be fatal. This could actually ending up killing the newborn, or as it could end up killing the developing fetus. So the way to counteract this, and this is another reason why prenatal care is so important, if you have this incompatibility, the woman will be given a drug called RhoGAM. So you can't have future pregnancies and this not be an issue. Again, typically not an issue with the first pregnancy, but it can be for any future pregnancies. And this conflict, like I just mentioned, is called Rh incompatibility, where the mom is negative, the baby is positive. Those are incompatible because the mother's body sees the baby as something foreign and needs to be attacked. And here's a illustration of this. Father Rh positive, mother Rh negative, baby positive. So this is incompatible with this. Mom sees this as something foreign and will start to develop antibodies to attack it. So the next pregnancy after this is where it becomes an issue. Alright, that concludes chapter 14, the components of blood. Again, like always, if you have questions, feel free to ask me or post to the discussion board.